Thank you everyone for joining us in our quarterly BGC Argo webinar series. Special thanks to OCB for hosting these webinar series and John Sharp for organizing this and the previous, I think, the webinar. My name is Takeshita, I'm an Mbari, and I'm going to do a quick introduction about BGC Argo and GO-BGC and any updates that may have happened in the GO-BGC program and over the past between the last webinar and this webinar, and then I'm going to hand it off to John, who is going to introduce our speakers for today. So quick reminder that BGC Argo is part of One Argo, which aims to maintain an array of 4,700 floats, and of which 2,500 will be core floats, so that's temperature, salinity, pressure, 1,200 deep floats, and 1,000 BGC floats distributed. This is the kind of the target distribution. And the number of 1,000 BGC floats was justified through various approaches, which is summarized in the Biogeochemical Argo Science and Implementation Plan. And if you haven't read this document, it's very, I think it's great. And so I highly recommend people reading this to kind of get the background of biogeochemical Argo and some of the justifications of why why the target array size is 1,000 floats. And I want to mention that BGC Argo data is a part of Argo, and the key feature is that all data are freely available. And then in real time, there are adjusted real-time data from BGC Argo, and then also delayed mode QC Argo data as well. And then so there are two main access points for BGC Argo data. One is at the global the GDAX. And we recommend users to use the SPROF files, which stands for synthetic profiles. And this is a file that have merged all the pressure grids from the various biogeochemical sensors into a single pressure axis. So it's a lot more user-friendly than trying to merge it yourself. And there are now monthly snapshots of both the whole Argo data set and also the SPROF data set as well. And so these monthly snapshots have a DOI. So whenever you use data for publication, make sure that you list that for in your references. And there are also BGC Argo toolboxes that help you extract data from the GDAX developed by John Sharp and Hartmut. And so in these toolbox codes are available in MATLAB, R, and Python, and the code is available on GitHub, and there are some tutorial YouTube videos that you can access through the GO-BGC YouTube channel. In addition to that, there are quarterly snapshots that contain predominantly SOCOM and GO-BGC data, and so these are delay mode QC data. And then the main difference between the GDAC and these snapshots is that we include the derived carbon parameters, which include PCO2, DIC, total alkalinity, and POC, so particulate organic carbon. So if you are interested in using these parameters, they are available for the subset of floats in the BGC Argo dataset. So as I mentioned before, the target of BGC Argo is to maintain 1,000 floats evenly distributed around the globe. So this is what 1,000 floats look like. And currently, in the current status of BGC Argo, there are 506 operational floats that have at least one biogeochemical sensor on them. And then this map shows of which 305 floats have at least 4+ sensors. And then that's actually 54 more floats since the last webinar. So this array is growing, but we can see, we are still quite a bit shy of our 1,000 float target array. And then I want to highlight a little bit about where recent floats have gone in through the GO-BGC and SOCOM programs. So this is a map of the GO-BGC float distribution. So in total, we have deployed 92 GO-BGC floats over the past 2 years. And then we have deployed 28 since the last webinar. And predominantly, these floats went into the Southern Hemisphere, highlighted in this white box. So these floats are only a couple of months old, but data are coming in. So if you're interested, please go check that data out. This map shows the deployment plans for GO-BGC and SOCOM over the next 6 months or so. And these open circles are floats that were recently deployed. So these floats down here that were not included in the previous map, these are SOCOM floats, but these are all in the water as well. And then so, and all these solid symbols are floats that are planned to be deployed in the next 6 months or so. So you can see that quite a few floats are going to go into the Indian Ocean and parts of the Southern Ocean and start filling in more of the Pacific Ocean as well. And all of these maps are available on the GO-BGC website under array status. In addition to the US contributions, there are also international commitments to BGC Argo. So international partners project that they are going to deploy at least 250 BGC Argo floats over the next 5 years. And just from the last ADMT, the international partners are planning to focus their deployments in these yellow squares around the world. And then so it seems like there is momentum and floats that are being deployed around the world, which is great. And finally, I wanted to end with a little bit of updates. So GO-BGC is hosting a science workshop in August or September. We're trying to finalize a date in the next few weeks, and it is most likely going to be hosted in Boston. And so please keep an eye out for the announcement of the workshop, and please register if you're interested. And this workshop, currently we're planning on making it a pretty hands-on workshop, kind of, learning, teaching people how to, use BGC Argo data and doing some preliminary analysis on that. And then the final 2 updates, these are just quick science updates, kind of what potentially can be done with BGC Argo data. These plots are courtesy of Ken Johnson. And then so the first one, this plot shows the oxygen distribution at various depths, so 200, 400, 300, and 600 meters by combining all biogeochemical Argo data. And you can see that you can map these floats are able to map out the distribution of the ODZ in the Eastern Pacific. And so so this is an annual snapshot, but soon the array size would be big enough to be able to constrain seasonal to annual resolution of the ODZ features. So if you're interested in looking at ODZ dynamics, this is an area that you might think about using float data. And then another update, another plot from Ken Johnson is we have recently deployed 5 SOCOM floats in the Ross Sea. And then so this is 75 to 80 south. And so these are the furthest south BGC Argo floats that have been deployed. And these were deployed relatively recently. So there's only a few profiles from each of these floats, but these sensors seem to be working. And then yeah, and as you can see, the depth is pretty shallow, so these floats park on the bottom and they come up. And so if you're interested in Ross Sea dynamics, these are great floats to check out. And also we have had, I think, 28 under ice floats surfaced this year, so we are starting to amass a lot of under ice BGC data as well. So that's another aspect that people can study using BGC Argo data. And I wanted to end with mentioning that all this data is freely available, and we encourage everyone to use the data. And with that, I would like to pass it over to John.
Thanks, Yui. Can I get a thumbs up if you can hear me? I know I was frozen earlier. Okay, cool.
It's great.
And I want to thank Yui for leading the team of organizers for this webinar, which includes myself, Channing Prend, Yvonne Huang, Lynn Talley, and Allison Gray. And we work with May and Heather from OCB, of course, so thank you to them as well. So the topic for this webinar, we're going to be hearing 2 different talks, is observing mesoscale and submesoscale processes with BGC Argo. And so our first speaker is going to be Lily Dove, who is finishing her PhD currently at Caltech, and will be starting a postdoc soon at Brown University to continue working on Southern Ocean submesoscale dynamics. And so, yeah, Lily is going to be talking about investigating ventilation at the submesoscale in the Southern Ocean. So take it away, Lily.
Great. Thanks, John. And thanks to all the organizers. I need to give a preliminary warning that I have a very excited cat who really likes being on camera. So you will probably see him passing by. But yes, I am extremely excited to be here. I love talking about BGC Argo and SOCM, and I am excited to tell you a bit about the work I've done with some collaborators on looking at submesoscale and mesoscale variability across the Southern Ocean. So you can see I've had wonderful collaborators in Drew Valwada and Allison Gray and my advisor, Andy Thompson. So when we think about the term ventilation, I think about exchange of water between the surface and interior ocean. And this takes a large variety of forms across the global ocean. One is, of course, the overturning circulation, which we are very familiar with, especially if you're a Southern Ocean person. This is where we, of course, have waters which have been sequestered for a long time, coming up to the surface, exchanging with the atmosphere, heat and carbon and oxygen, and then getting evected away. And there are lots of ways this happens, but that's kind of the big picture ventilation in the global ocean. But ventilation also takes other and smaller forms, one of which is through mesoscale stirring. So if you imagine a mesoscale eddy, and you know that it kind of has a bowling shape, a bold shape, and that you have isopycnals or density surfaces tilting up and down. That's another way. If you get stirring along those density surfaces, that's one way you can get ventilation or exchange between the surface and interior. And then we additionally have even smaller scales, so submesoscale subduction and entrainment of waters. So I'm going to be focusing on processes at the submesoscale and mesoscale, but I should give the preface that it's really hard to separate out these scales. It's definitely more of a spectrum than a clear definition. So just keep that in mind as I'm going forward. But one way we do attempt to separate out the kind of mesoscale versus submesoscale scales is looking at spatially. So this is a ocean color image from Drake Passage, looking at pointing out a big, nice, beautiful mesoscale eddy spinning around phytoplankton. And that's on the order of 100 kilometers, probably smaller because it's the Southern Ocean. But we also have filamentary structures at the submesoscale, so orders 1 to 10 kilometers. And one way that these submesoscale features can play a role in transporting water from the surface into the interior ocean is demonstrated on the right here. This is a really beautiful image from one of Marina Levy's recent papers. And plotted on the top is a distance along across a front, a submesoscale front. So like looking this way, then a depth on the y-axis. And in the color is chlorophyll. So brighter yellows means more chlorophyll. And you can see this interesting patch here that's below the mixed layer. But if you plot this in density space, so our y-axis changes to density, you can see that that water was actually transported along a density surface. And this is a signature of submesoscale subduction of waters from the surface into the interior along density surfaces. And I should say that the Southern Ocean, which is where I'm going to be focusing today, is really preconditioned to submesoscale motions. There are a lot of things that precondition a region, but some of them are persistent wind forcing aligned with fronts, which we get regularly in the Southern Ocean. There are also vigorous eddies, which lead to strong lateral buoyancy gradients or density gradients, which I'll be talking about a lot more later on. And finally, the Southern Ocean has reduced stratification, which allows surface waters to have low potential vorticity values. And all of these things combine to precondition the Southern Ocean to submesoscale motions. And I should say this beautiful figure is from Sarah Gilly and others' chapter in the recent ocean mixing book. So my question that I decided to go after was looking at what scales of variability can we observe in a high eddy kinetic energy region of the ACC. So to put us all on the same page, the ACC is the dominant eastward flowing current of the Southern Ocean. And the Southern Ocean is kind of divided into high energy regions and low energy regions. Of course, that's also a spectrum, but these high energy regions are a result of interactions with topography at the seafloor, which eventually lead to high energetic stirring at the surface. And so when we look at one of these regions, this is Africa here, down here is the Antarctic continent, and this was a GO ship line that went from Durban south and deployed BGC Argo floats or SOCM floats along the way. Yes, BGC Argo floats. And we also deployed a couple of sea gliders alongside this, alongside one of these floats. In this red box is what's plotted on the right here. And you can see what's plotted in the red is the value of eddy kinetic energy. So darker red colors mean high, more energetic, more mesoscale eddies stirring up the ocean. Lighter colors mean less energetic regions. And they're all overlapping, but you can see we have three different things in the water here. We have one BGC Argo float in the green dots, and then we have two sea gliders following this float. For those not as familiar with sea gliders, they are a piloted remote tool or autonomous tool. And so we were able to track float slightly. You can see that in the high eddy kinetic energy region, things got a little more complicated. It was harder to track the float than we expected. But near the end, when we were in this lower eddy kinetic energy region, a calmer area, more quiescent, we were able to track the float more effectively. So having these two different tools at our disposal is extremely useful. And I'm going to talk a little bit about some of the results we saw and then how we applied that to the full Southern Ocean. But before I can do that, I need to introduce you to the concept of AOU, which is the difference in saturated oxygen and measured oxygen. I'm sure that Shuangling's talk, which is the next talk after me, will go much more into oxygen. So if this is piquing your interest, be sure to stay tuned for that. But I'm looking at AOU, which is, again, this difference between measured saturated oxygen and measured oxygen. And saturated oxygen is set by set by the temperature, and you can imagine it as the concentration of oxygen that water would have if it was in contact with the atmosphere. So waters that are closer to the surface are more likely to have low AOU, meaning their measured oxygen is close to their saturated oxygen content. And this is because this measured oxygen has recently been exchanged with the atmosphere. However, in this simplified case, waters that are at depth are more likely to have high AOU or are less recently ventilated. And that's because your measured oxygen is decreasing because it's being used up in respiration. But you can imagine if you have some way of getting low AOU waters from the surface to depth, potentially along density surfaces, you can get values of low AOU water at depth that you wouldn't necessarily expect. And so this is what we decided to look at, and we are using this value that we call delta AOU. And what this is it's the difference between the AOU at a set depth, which we define as delta H, and the AOU at the base of the mixed layer. And we expect this AOU at the base of the mixed layer to be close to 0, just by definition of AOU. But we did this correction just in case. So what I'm plotting here results from the sea gliders. And what is plotted in purple is from the low EKE region, so that downstream, more quiescent region, and in green is that from the high eddy kinetic energy region, so right when we deploy the gliders, kind of directly into the polar front. And on the x-axis is the delta AOU at a given delta H of 200 meters. And on the y-axis is just kind of the frequency of seeing that observation. So what stands out to me is that the low EKE region has this kind of well-defined Gaussian distribution, but has average lower AOU at 200 meters than its high EKE region counterpart. And remember, this is a set depth below the base of the mixed layer. The mixed layer depth didn't change that much throughout the deployment. So what we think we're seeing is a real difference between ventilation that's happening or movement of water between the surface and the interior ocean between the high and low EKE regions. And I'm going to get into this much more. But the gliders gave us the opportunity to begin pulling out some of the processes that might lead to this distribution. So what's plotted here is on top, you can see the streamlines of a mesoscale eddy. And in this curtain plot are the glider measurements. And each of these dots, the green, the blue, and the purple, represents one float profile. So this float was traveling around the eddy, and our glider did a bit of a cross across the eddy into the center and then back out again. And what really you can see is that there's this low AOU anomaly at the edge of the mesoscale eddy that is captured by the glider measurements, but is not necessarily captured by the float profiles because it's happening on such small spatial scales and also small temporal scales. We imagine that this process probably happens on the order of days. But you can see that it's hard to tell with this kind of plot, but this is low AOU water. So recent water that's recently been at the surface and might hold high chlorophyll content. That's been brought down to about 300 meters. And this black line represents the mixed layer depth. So that is significantly below the mixed layer. And we expect that this is probably kind of permanent entrainment of surface water out of the surface ocean. So the question I had was, wow, this is really cool. We found this in one specific region of the Southern Ocean. Can we expand this from one single high energy region to the full Southern Ocean? And the answer is yes, thank you to SOCM and BGC Argo. So what's plotted here is my definition of the Antarctic circumpolar current. And in the red dots are the number of float profiles with oxygen that were available at the time we did this study. And you can see that there are some regions with more float profiles with oxygen than others, but that overall we have pretty good coverage of the region that we wanted to look at. On the right is a plot again of this eddy kinetic energy. This is log 10. So these blue values represent high eddy kinetic energy. These orange values are low eddy kinetic energy. And to put it into context, this region here, the Crozet Plateau, is the region we were looking at before with the gliders. So I wanted to look at not just the Crozet Plateau, but also Kerguelen and Campbell and Drake Passage, and compare those high eddy kinetic regions to their low eddy kinetic region counterparts, and see if this message we found about ventilation being stronger or more active in high eddy kinetic energy regions held across the full Southern Ocean. So once again, I'm using this kind of definition, and this is again looking at histograms. So on the x-axis is delta AOU, y-axis is probability, and you'll see the delta H value written up here. And you can see the blue and the orange curves represent high and low eddy kinetic energy region respectively. So 100 meters below the base of the mixed layer, you can see that our curves in the high and low EKE region are pretty similar. There's not too much difference in the AOU at that depth. But things get much more interesting when we go a little further down. So at a delta H of 400 meters, you can see kind of that same signature we'd seen before. So this is across the full Southern Ocean. You can see that we have this values in the low EKE regions of higher delta AOU, suggesting older waters, less recently ventilated waters. While in the high EKE region in blue, we have a lower median and also this interesting double peak situation going on, but lower average delta AOU values. But when we get even further down, we see that this pattern goes away. That at 700 meters below the base of the mixed layer, our two curves come together again, and there doesn't seem to be a difference in AOU at that depth. Another way to look at this is to plot the median delta AOU on the x-axis and then look at delta H on the y-axis. And you can see, again, near the surface, there isn't too much difference between high and low EKE regions. Again, at 700, 800 meters, there's not too much difference, but in this intermediate zone, there's a lot of difference between delta AOU. And again, pointing towards that in high eddy kinetic energy regions, you have a lower delta AOU, suggesting more recent ventilation or more active ventilation. So this is all very interesting that we have this we have an understanding that there's probably more active ventilation happening in high eddy kinetic energy regions. But the question becomes, do we know what processes are leading to those distributions that we are observing? And so this led to me attempting to pull out some process about processes on the sub-mesoscale to mesoscale continuum from the floats about what's setting these distributions. The number one process that we decided to look at was along-isopycnal stirring. So this is just to give us a simplified view of what I'm talking about when I talk about along-isopycnal stirring. If you have for lack of a better word, a boring ocean, you would expect you have low density waters at the near the surface and high density waters at depth. And you'd have new waters, so low AOU waters and at the surface, and old waters at depth. And we know that waters like to stay at their given density. They just like to stay on their density surface and do adiabatic stirring. And these gray lines are representing those isopycnals, or lines of constant density. But we know that in the Southern Ocean, things aren't this simple. In fact, we have our isopycnals are tilted as a result of the geostrophic flow of the ACC, which tilts our density surfaces upwards, upwards towards the continent. And so that means that while we still have this gradient of new waters to old waters from the surface to depth, we instead have these tilted isopycnals with low density on one side and high density on the other. And because waters like to stir along their given densities, we can get a long-isopycnal stirring of tracers, and in this case, AOU. So in this case, we could get low AOU waters coming from the surface stirring alongside old waters. And this is one of the processes we suggest is happening more regularly in high eddy kinetic energy regions as a result of the mesoscale eddies present. And the way we decided to look at this is using a long-isopycnal variance of AOU. So what's plotted here is that AOU variance. So basically, do we have a larger range of AOU on a density surface or a smaller range of AOU on a density surface? And on the y-axis is those given density surfaces. And what you can see is that in the high eddy kinetic energy regions, you have larger variance of AOU on density surfaces than in these low eddy kinetic energy regions. And interestingly, that difference goes away in denser waters, but it is very prevalent in these intermediate depths, especially waters that are aligned with intermediate water masses. The other process we wanted to investigate was looking at submesoscale vertical velocities, or transport, pretty rapid transport of waters from the surface to the interior ocean. And to investigate this, we needed to pull in some satellite data. The data we looked at were of finite-size Lyapunov exponents. I know that's probably a term not as many people are familiar with, but these are values that are derived using a remotely sensed sea level anomaly field. And the values tell us about the strain experienced by a Lagrangian particle in a velocity field. So I'm just going to take a screenshot here. So you can see that we have, in this GIF, you are distributing tracers in a field of spinning eddies. And you can see that depending on where the tracers are originally set, they're going to have different trajectories and ultimately experience different values of strain. And so in this example, these particles that were set in the red region, kind of in the middle of this eddy, they're spinning around for sure, but they're not experiencing a lot of strain. They're not being stretched apart really rapidly. And so this is what we consider a low magnitude FSLE. On the other hand, these black particles are being placed kind of in this saddle point and are being stretched rapidly apart. And you can see that this happened over the order of just 10 days. You got transport of particles degrees of latitude apart from each other. And so this high strain results in high magnitudes of FSLE. And the reason I brought FSLE into this is that FSLE has been found to be empirically correlated with lateral gradients of buoyancy at the submesoscale, which we know are a precursor for submesoscale vertical velocities. This is work that Leah Siegelman at Scripps has really pushed forward. But you can see on the right that in the red to blue are colored FSLE values. And that's what I just described. That's that satellite measured value. And then in the green is this horizontal buoyancy gradient. You can see that region, there are spots where the FSLE is high, are also regions where this lateral buoyancy gradient is high. And one way to visualize this is plotted on the left here. So this is that lateral buoyancy gradient. You can see these strong these isopycnals coming to the surface, and you have a strong lateral buoyancy gradient. And at that region, you can also see you have chlorophyll being transported to depth along those density surfaces. And ultimately, because the ocean doesn't like these density surfaces to be facing up and down, it wants them to be flat. Ultimately, when the ocean spins out this, this lateral buoyancy gradient, that chlorophyll is going to be trapped below the base of the mixed layer. So this is the process that we are attempting to get after. It's difficult to really put values to this just yet, but what we can see is that regions of high eddy kinetic energy, once again in the blue, as compared to the low EKE regions in orange, have stronger FSLE values or greater strain and potentially stronger fronts, sub-mesoscale fronts, than the low EKE regions. And you can see that plotted here. Strain increases to the left on this plot, so greater magnitudes of FSLE. And your probability is on the y-axis. And so in our high EKE region, we have more values of this stronger strain and stronger fronts than these low EKE regions. So I'm going to wrap it up there. I am always happy to talk more about this work, so please reach out to me. But what we've done with this work is shown that ventilation is localized to eddy kinetic energy hotspots, which are downstream of flow topography interactions in the Southern Ocean. I didn't talk too much about that second part, but there's a lot of work out there that describes how these high EKE regions in the Southern Ocean actually come to be. We believe that this ventilation is primarily controlled by processes at the submesoscale and also the mesoscale. And finally, this requires more observations to constrain, not just at the largest scale, but also through more floats and through more glider observations and through other observing systems. So I'm going to leave my summary up for you. If you're interested in reading more about this work, I highly encourage you to go check out some of these publications. And I, once again, am really grateful to be here today. Thank you.
Awesome. Thank you so much, Lily. I will quickly queue up our next talk here and invite anyone who has questions for Lily to add them to the chat or save them for the end. I think we'll have a few minutes to ask questions at the end of the hour and to maybe have a quick discussion about how the emerging GO-BGC array might kind of help us answer some of these questions about mesoscale and submesoscale processes. All right, so I'm sharing a window here. Our next talk is from Shuangling Chen. It's a recorded talk because I believe it's the middle of the night for her at the moment. She is a researcher at the Second Institute of Oceanography in China. And I'm happy to have her talk because the two of us actually overlapped for a couple of years when I was in grad school at the University of South Florida. And so she's going to be talking about a similar topic as we just heard from Lily, episodic oxygen injections, but this time not the Southern Ocean, the North Pacific. And so I will play this right now and let me know if in the chat if the volume is okay.
Okay. Hello, everyone. I'm Shuangling Chen from the Second Institute of Oceanography in China. Thanks, John, for the invitation for the BGC Argo webinar. It's really my great honor. The talk I'm going to give is on the episodic oxygen injections we observed from BGC Argo flows. Professor Mark Wells, Ruixin Huang, Huixie Jingyan, and Fei also involved in this work. Here I acknowledge NSFC SIO, SOED, and other institutes, as well as the BGC Argo data agencies for supporting this study. First, oxygen really matters. Phytoplankton is a key producer of oxygen in the ocean where photosynthesis. Oxygen is critical to the health of our planet. It supports around 50% of the oxygen we breathe every day. It affects the cycles of carbon, nitrogen, and other key elements, and is a fundamental requirement for the marine life from the seashore to the greatest depths of the ocean. However, studies show that oxygen concentration in both the open ocean and coastal waters have been declining for at least the past half-century. While the decrease of oxygen could have a wide range of biological and ecological consequences. For example, it can cause major changes in ocean productivity, biodiversity, and biogeochemical cycles. While analysis of the direct measurements at sites around the world show that low oxygen zones in the open ocean have expanded by several million square kilometers. And hundreds of the coastal sites now have oxygen concentration low enough to limit the distribution and abundance of animal populations and alter the cycling of important nutrients. While in the open ocean, global warming is considered as the primary cause of the ongoing deoxygenation. Or specifically, the rising seawater temperatures decrease the solubility of oxygen, but it increases the rate of oxygen consumption via respiration. But in addition, the increasing stratification and the decreasing ocean overturning circulation also reduce the introduction of oxygen from the atmosphere and surface waters into the deep oceans. Therefore, any processes that could alleviate the poor oxygen conditions in the ocean interior are important to know about. So here we focus on the eddy-driven subduction. It's a physical process on meso and submesoscale scales that transports surface waters into or below the thermocline. For the intensification of fronts within the eddy flowing field could generate the downward velocities along the sloping isopycnal surfaces that create subsurface intrusions of the surface waters. While the resulting subduction leads to the episodic injection of the surface waters to the depths. While studies show that eddy-driven subduction are mainly concentrated in the hotspot of high eddy activities along the perimeter of eddies with a scale of around 1 to 10 kilometers. Well, the subducted waters usually have a high concentration of oxygen and organic carbon. As such, the eddy-driven subduction could serve as a potential pathway for oxygen and carbon transport into the deep ocean. But right now, many studies have demonstrated that eddy-driven subduction could export around 20 to 50% of POC into the deep ocean. That's an important mechanism for carbon dioxide removal that we care a lot nowadays. However, our knowledge on how much oxygen could be injected into the deep sea is quite limited, and this is our general motivation of this study. So to detect the subduction signals, we need biogeochemical profiling measurements around eddies, which is quite difficult for the traditional platforms. For the recent development of the novel BGC Argo platforms really provide a powerful tool for such measurements. Well, in this study, we focused on the Western North Pacific, where meso and submesoscale eddies are very popular in this energetic ocean environment. And we analyzed over 7,000 BGC Argo profiles collected between 2008 and 2019. Here on the left is the Argo trajectories, and on the right is the data density in a 25-degree grid. Well, when a BGC Argo float passed through a parcels of water injected from the mixed layer, it could capture coherent anomalies in AOU and sparsity as well as other properties, which are distinct from the surrounding waters. Here highlighted in yellow are the subducted waters in patch. Well, sparsity is a water mass marker derived from temperature and salinity. It's a water property similar to density, but perpendicular to density. Following the published studies, here we use the anomaly in AOU and sparsity to identify the subduction patches at depths. And these patches are indicators of the subduction events occurring in the vicinity. And we developed approaches to quantify the anomalies and identify the subduction patches for each profile. Here, I'm not going into much details on the method about the quantification, but if you are interested, please refer to our paper for more details. And here is one example of the eddy-driven subduction captured from one Argo float. The left panel shows the trajectory of the flow, and the right panel shows the profiling time series of AOU and DO. Clearly intermittent patches of elevated DO and lower AOU are distinctly visible at depths of around 450 meters. And the thickness of this subduction patch is around 50 meters. Following the procedure we developed, we processed all the data profiles collected. It's found that in total, 4% of the profiles were identified with subduction, and over 50% of the subduction patches were found in the crucial Oyasho extinction region, where the eddies are most popular. In the North Pacific, a study showed that the depth of the permanent pycnocline was around 450 meters in the subtropical region and 300 meters in the subpolar regions. So based on the depths and geolocations of the subduction patch we identified, we found that over 70% of the subduction patch were below the permanent pycnocline, and some of the subduction injections could go even deeper than 600 decibel. And these deep oxygen injections should mean a lot to the organisms living in the deep sea. And we also investigated the temporal distribution of the subduction patches in terms of the integrated number of patches, AOU and DO along the day of the year. It's found that 70% of the subduction occurred between March and August, peaking in March. And we think that the more energetic winds and the accumulated strong heat loss in winter probably contributed to the peak of subduction observed in March. And in general, the oxygen concentration of the injected waters is around 20% higher than the surrounding waters. To further examine how much oxygen was injected into the deep waters, here we quantified the oxygen inventory for each subduction patch by integrating the anomalies in dissolved oxygen. That is the shaded area in the profile. So we did two kinds of quantification. On the left, it's based on the anomaly referring to the background of the dissolved oxygen, and on the right is based on the maximum oxygen anomaly within the subduction patch. So it's a maximum potential of the oxygen inventory. In total, the oxygen inventory within this subduction patch were estimated to be around 60 to 150 gram oxygen per square meter. However, it should be noted that based on the Argo data alone, we can't capture the big picture of the horizontal extension of the subduction because of the Argo float only captures a snapshot of the subduction process. So as such, we can't estimate the exact volume of the subducted waters. While considering the decline of the oxygen, particularly in tropical and subtropical zones, and the strong eddy activities in the crucial Oyasho extension, we think that the injection of oxygen by eddy pumping in this area should be a significant input of oxygen into the deep ocean. Here is a brief summary of this study. So in general, based on the chapter of the BGC Argo data profiles we collected in the Western North Pacific, we investigated the spatial and temporal characteristics of the eddy-driven subduction, as well as oxygen inventories within these subduction patches. However, subduction is a four-dimensional physical process, while the Argo float only captures the snapshot of this process. So next, we need to work together with the ocean modelers to resolve the full picture of the subduction induced by meso and submesoscale eddies. Right now, these mesoscale and submesoscale processes are not well considered in most of the current biogeochemical models. So further studies are needed for a better understanding of the oxygen transport to the ocean interior via this subduction. So due to the time limitation, I'm going to stop here. Please refer to this article for more details if you are interested, and please feel free to reach me if you have any questions. And I also put my webpage here. My research is mainly on ocean color remote sensing and ocean carbon cycling, focusing on the air CO2 fluxes, nutrients, and new production. Right now, we are taking advantage of both ocean color remote sensing and the BGC Argo flows on the four-dimensional ocean observations to study the ocean carbon cycles. So please do let me know if you want to collaborate on any of these topics. With that, I'm going to stop. Well, thank you all very much.
All right. And yeah, I'll just reiterate that anyone who is interested in Shuangling's work and or has any questions for her, please feel free to send her an email. I'll put her email address just in the chat. So let's have it. Okay, so it looks like Lily has done a pretty great job of answering all the questions that have been posed in the chat so far. I don't think that there are any loose ends right now. So I'll just invite anyone else who has a question and wants to, if anyone wants to unmute and ask a question, please go ahead. Or Lily, if you want to expand on any of your answers, I will invite you to do that as well.
I had a question. This is Yui. So I guess this is for, a question inspired by the spiciness, but Lily, can you use spiciness as a tracer in the Southern Ocean to look for these subducted filaments? I just don't know if there's enough of a, spice signature in the mixed layer versus the sub mixed layer, because in your answer you say that, yeah, you need a passive tracer, and spiciness is kind of a passive tracer. If you ignore mixing, it is diapycnal mixing, it is a passive tracer. So I was wondering if you have attempted that before.
Yes, it's a great question, and it's absolutely a really useful tracer. The challenge with it, with Shuangling's work, you can see that when you're comparing directly to the surface above you and then below, you're able to, you can kind of do that like correction out for the surface. The challenge is, if you're looking like across a large region, AOU or spice varies greatly with latitude across the Southern Ocean. And so that's why we used AOU. It's because AOU is like corrected to 0 at the surface. And so, yes, you can use it, I would say, in like localized or like looking at like individual subduction events. And it's really useful in that context. I'd say it's much harder in like larger like regional work, especially if you're looking across fronts, especially.
Maybe a question related to that, Lily, thanks for a really nice talk. Is there a signature in any of the other BGC tracers that can be useful, or like, especially for trying to tease apart like biotic versus abiotic effects?
Yeah, this is something that I think is very interesting, but will be the first to admit I'm a little I'm a little scared to touch chlorophyll, just because I know you can make a whole career out of that. But yes, absolutely, there should be and are often chlorophyll signatures associated with recent subduction. But not always, and why that occurs and where and when is especially interesting. And so, yeah, the question, at least to me, becomes like, okay, we're seeing recent ventilation of oxygen, which you can take the next step and tie that to carbon, but until you start actually tying that to chlorophyll or even backscatter, you don't know if it's like a one-to-one, if the ventilation that's occurring is how much carbon it's actually taking with it. So it's definitely a question with a lot of room for biological and physical oceanographers to like work closely together on.
All right, well, I see people dropping off the call as we're past the top of the hour. So I just want to say thank you again to both of our speakers and Yui as well. I'll give a round of applause, and I'm sure everyone else will also. And thanks, yes, stay tuned for a few months from now when we'll be hosting another of these webinars. Thanks, everyone.